Okay, let's go ahead and get started. So our first lesson that ended just barely was cell respiration. And what this lesson did is it started us out with understanding what's happening in any combustion reaction. And it's just a burning candle in this case. And what we're seeing is that the burning candle goes out. Why does it go out? It goes out because the uh, the oxygen in the jar is consumed. And when the oxygen in the jar is consumed, there's nothing left for that reaction to keep happening, right? And the reason why that is because oxygen is the final electron acceptor for the combustion reaction, right? And the reason why it's such a explosive reaction is that we're releasing all of the energy at once. So if we're imagining high energy electrons in the paper, right, that's going all the way down to a low energy state on the oxygen. And so that change in the energy state results in heat being released all at once. If we contrast that to the uh, the fluorescent stars that we looked at in our chemistry lesson. The fluorescent star lets the energy out a little bit at a time, stepping down more like this as it moves down the energy levels. So our cells, we are going to do a combustion reaction, but we don't want that combustion reaction to be all at once, right? That would release all the energy of the food that we eat at once and we get burning, right? We'd have way too high of temperatures. So we want to do this, but we want to do it step by step and slowly release the energy. And um, this kind of gives us an idea of where the energy is coming from, right? It's coming from breaking bonds. When those bonds reform, energy gets released. We're also going to get high energy electrons out of this that we're going to use later in this process. We take those high energy electrons, we put them into a molecule called NADH. And every time we break subsequent bonds in our sugar molecule and reform new bonds, we get more high energy electrons. So um, that's kind of the idea behind this process. We're also just a moment. Sorry about that. Sorry about that. So what we're going to see is in the cell respiration process, right? As that energy gets released as it drops down from one shell to the next, we're going to use that energy to cause active transport to occur. Instead of going from high concentration to low, we're going to force hydrogen ions to go from low concentration to high. And that builds up a gradient outside. And then those hydrogen ions will want to come back into the center, the matrix of the mitochondrion. And in order to do that, we're going to allow it to pass through, but it has to pass through this special channel protein. So facilitated diffusion here. So as it goes from high concentration to low, it's facilitated diffusion. And this special protein acts kind of like a turbine. It's going to allow the water to pass through, not the water, sorry, the hydrogen ions to pass through. But as it passes through, it's going to turn the turbine. And there's this special protein, and that mechanical energy is going to be converted into chemical energy, into ATP, which we talked about last time, is that temporary storage that our cells need, molecule that our cells use to use the energy whenever they need it. So that is kind of the idea that Dr. Jensen talks about fully in the video. And so I don't want to go through everything in the video unless you have questions on it, because she does a great job explaining that and giving us a visual to go with it. But this is the general idea that we're doing, right? We're taking the energy from the bonds of the glucose molecule, and we're getting high energy electrons out of it. And those high energy electrons are used to generate ATP. Are there any questions about the cell respiration lesson that you'd like to go over? Okay. All right. Photosynthesis. So in this one, we're seeing some laboratory experiments when we look at this essentially how much photosynthesis is occurring in a plant based on the type of light that is being used. And hopefully what you can see is that the rate of photosynthesis is impacted by the color, and certain colors work better than others. And what you should have seen is the color that works best is those that are blue, purple, and the colors that work worst are the green. And if we think about why that is, right? Uh, when we look at something and we see light reflecting off of it, we see the color of light that it's not absorbing, but the color of light it's reflecting. So when we look at a green plant, all the colors of light are hitting it. White light has all the colors. The colors that are bouncing back are green light. And the reason why they're bouncing back is because they're not being absorbed. If they're not being absorbed, they can't be used to build sugars through photosynthesis, right? That's the light that it's giving off, that it's reflecting. And so that's why green light is the worst color of light for photosynthesis. Some key things that we found from these experiments is we saw that the oxygen ends up being the oxygen that plants release that we breathe in. Oxygen is coming from water, and that the carbon dioxide oxygen is what ends up in the sugar molecules. And as Dr. Jensen went through the photosynthesis lesson, you should have seen that how we've put those two oxygen molecules or oxygen atoms into this process, right? That the oxygen that's in water is released because the electrons are being ripped away from the water molecule to be replace the electrons that are going through photosystem 1 and photosystem 2 in this process. So water is in this chemical reaction lost, destroyed, right? Ripped apart. And oxygen is then released from that process. And then the carbon comes into the process, right? The carbon that's with in CO2 and the oxygen that goes with it, that oxygen ends up in the sugar because in the Calvin cycle, right, we have rubisco, that enzyme that grabs the carbon dioxide and takes it through the Calvin cycle, and the energy that's generated from what we call the light dependent reactions. I believe Dr. Jensen calls them the light reactions in her video. These reactions provide the energy for the Calvin cycle to function. So that's powering this system. And then the Calvin cycle is where we get the first a 3-carbon sugar, G3P built, and then those two will be linked together to make the 6-carbon glucose molecule. So we have the light reactions or the light dependent reactions, and we have the dark reactions, which is the Calvin cycle, the light independent reactions or dark reactions that are building the sugar. So the energy is coming from the light reactions. The sugar molecule gets built by the dark reactions. Water's consumed as the reactant in the light reactions, and then carbon dioxide is used up in the dark reactions, and that they're all tied together, right? And then we produce the oxygen, which is kind of the waste byproduct, and the sugar molecule, which is really what the plant is interested in. So those are kind of the big things that we needed to learn from this lesson. There are some things that can happen that are in rare circumstances, which Dr. Jensen talks about in photorespiration and fermentation. But again, the videos are better than I could probably explain here, so I won't go into that